Good afternoon and welcome to part 2 of our presentation on using the Seahorse XFp. Today is February 10th, 2023, and this is the mitochondrial damage modeling and measurement group meeting. At the last meeting, I talked about the theory and concepts behind the Mito stress test and ATP rate assay used in conjunction with the Seahorse XFp that we have here on campus. Today, I'm going to be talking about practical tips and techniques that you will need to know and will be useful to you when you use the Seahorse XFp. Specifically, these are things that are not necessarily indicated in the manufacturer's protocol and documentation, but that are nevertheless important to doing these assays successfully and getting a high quality result. And this presentation also contains information specific to using the Seahorse XFp that we have here on campus. I'm going to talk about these tips and techniques in the same order that you would do them were you doing an actual assay. So specifically, I'll be talking about things you need to know before you perform these assays, things you'll need to know when seeding cells, treating cells for this assay, preparing the cartridge that is used to measure pH and oxygen consumption, things you'll need to know when making the media and diluting reagents that accompany these assays, washing and preparing what are called the mini plates that you culture your cells on, transporting your plates to the Seahorse XFp here on campus, loading the injection ports, normalization methods, generating assay reports which contain the final data that you'll be getting, as well as the cost of using this and a few final notes. Familiarize yourself with the Seahorse mini plates. These are the specially designed plates that the Seahorse XFp can hold and can measure your cells in. Now, these plates are not coated, they're not tissue culture treated, so it's hard for cells to adhere to them. However, if you use a coating on your plates, you can coat these plates yourself. I believe you can use both collagen and Matrigel as just two examples. I think that's actually why they're untreated. Poly-D lysine coated plates are available as well if you want to buy those from Agilent. The surface area of these plates is about 40% of the surface area of the well that you would have in a 96 well plate. So you'll want to correct for that accordingly. Now, Agilent says these plates can hold up to 250 microliters. They can actually hold about 500 microliters. That fills them all the way up to the top, but you can't fill them up that high when you're running the assay, but you can culture cells that way. So if you have a longer treatment and you don't want to change the media, or your cells are at a low density when you're seeding them in the plate, you can add up to 500 microliters of media if that is necessary for your purposes. On the right here in this image, you can see exactly what the plates look like. The plate that's in this image here, the image on the far right, that's actually a utility plate, which I'll be talking about later, but it's identical to the mini plate. So these are what they look like. Now, there are 8 wells in the mini plate. However, you need to leave the top and bottom wells, that's well A and well H, empty for calibration purposes. So that means you have 6 wells that you can assay. Typically, you can do either triplicates with 2 conditions. So in this case, you'll have your control and experimental 3 each. That's very often what's done. If you have another format you want to use, you can actually edit the template and load it onto the XFp so that it will be able to take those measurements and the format that you specify. One very important question here is how many cells you want to seed in each well. For our cells, trabecular meshwork cells, I recommend 10,000 to 40,000 cells per well. It varies heavily for us. We're one of the only labs who uses this cell type. So I might recommend starting out with 10,000 cells, but if your cell type is one which has been used before, you can look up to see what previous publications have done, and that would be a good starting point to make sure you have enough cells to stay within the measurement range of the instrument. It's also possible if you do duplicates to have 3 conditions. So you can have control experimental condition 1 and experimental condition 2. And so you can have 3 conditions in this way. I have already created a template for this for both the ATP rate assay and the Mito stress test that has already been loaded onto the Seahorse XFp that we have. If you want to do this, let me know and I can give you a little bit of extra guidance on how to find that and to use it. One of the things that you might do when you are seeding cells is you might actually seed cells into well A and well H, just out of habit, because most multi-channel pipettes have six pipette tips, 8 pipette tips that you use. But if you happen to do this by accident, you can kill off the cells in there and rinse them out by incubating them in water or just exposing them to air. So that's a quick tip if you found you made that mistake and accidentally let it sit overnight or something like that. One question you might have is, should you treat your cells and then seed them and assay them? So for example, should you have 2 96 well plates with your control and your treatment, incubate it for 48 hours and then seed it? Or should you seed the cells in the mini plate and treat them there? I recommend the second option. I recommend seeding the cells in the mini plate from one, say 90 millimeter dish at the appropriate density, letting them attach overnight, and then treating them for however long your treatment is. I think this enables more consistency in seeding density. It's certainly true that maybe your treatment causes the cells to proliferate more or less in a given well, but overall, I believe this offers the best consistency and the best quality results. So after you've seeded your cells and you've had your treatment, you are ready to action, and you've reserved the Seahorse XFp, you're ready to actually perform the assay. The day before you perform the assay, there are a few things to do. You'll want to turn on the Seahorse XFp to allow it to reach 37 degrees Celsius, and you'll want to do the same for the non-CO2 incubator in the stem cell core. You can do this the day of. I believe the most important criteria for the XFP is that it reaches 37 degrees temperature. I don't know how long this takes. One time I forgot to turn it on the day before, and I turned it on, 5 hours before, and that seemed to be okay. But turning it on the day before is definitely the best way to avoid any problems. Now, in addition to the mini plate that I described, to perform assays, you'll need to use something that is called a cartridge from Agilent. These components here, which are shown in green from 2 different angles, have an oxygen and pH sensor on them. I haven't been able to figure out exactly how this works, but I do know that there's a compound on the end of the sensor, and you can see it down here, these probes, that reacts with both with acid levels in the media and also with oxygen, and it produces a color change that the instrument is measuring. That's how it seems to work. So these cartridges are a one-time use thing, but that does mean you don't have to worry about cleaning sensors or anything like that. Now, before you do the assay the day before, you'll want to open the cartridge packet and add deionized water to the cartridge to hydrate it. In addition, the plate has, in order to avoid edge effects, it has wells on the side that you can put water in to keep the humidity level up when you incubate it. So that's useful as well. If you're doing 2 assays in a given day and you want to avoid confusion between which plate you've hydrated and which has the calibrant in it, which we'll discuss later, you could say not add any water to the motes when you hydrate it the night before, then add it when you add the calibrant. In addition, Speaking of the calibrant, this is a component that you'll use the day of the assay, but you want to allow it to reach 37 degrees Celsius. So you'll want to put the calibrant liquid in a non-CO2 incubator as well to allow it to warm up. After you've added the water to this cartridge, it needs to go in a non-CO2 incubator as well. So in summary, put the calibrant tube at 37 degrees, non-CO2 to warm up, add water to the cartridge, do the same thing, 37 degrees, no non-CO2, 24 hours the day before you do the assay. So moving on to the day that you're conducting the assay, generally speaking, the first thing you'll need to do is prepare the Seahorse media that goes with the assay. This is specially supplied from Agilent, and it's necessary in order for the assay to be conducted. It has special properties. It has no phenol red. It's pH balanced at 7.4, and it has 5 millimolar heaps, among other things. You can see it here. In addition, you want to protect this media from light. I have it exposed to light here just for demonstration purposes, but it is light sensitive, so you'll want to make sure you do that. You'll want to add, for one plate for one assay, you'll want to add 100 microliters of pyruvate glucose and glutamine, all supplied from Agilent, to 9.7 milliliters of media supplied from Agilent. The morning that you do this, you'll want to then put this in a non-CO2 incubator to allow it to reach 37 degrees as well. You cannot prepare this the night before. That's not recommended. You want to prepare it the day of, and you want to get it to 37 degrees non-CO2. Once this has reached 37, once this media has reached 37 degrees in a non-CO2 incubator, you're going to want to rinse your mini plate that has your cells in it. So you've got your cells in a CO2 incubator with the media you cultured them in under treatment. And if you're doing the ATP rate assay, you'll want to wash this 3 times using the Seahorse media that I mentioned. You also want to make sure you conduct the wash thoroughly to remove your phenol red and any relevant buffer. So for example, and for our purposes, we use 15 millimolar heaps, which is 3 times what the Seahorse media uses. And since pH measurement is such an important component of this assay, you'll want to make sure you have fully removed the media you're working with before that has FBS and all that other stuff, and make sure you have only the Seahorse media in there. You can rinse this just in a standard way with a multi-channel pipette or with an aspirator if you want. However, when I've tried those methods, I've found I've actually lost a lot of cells because they don't adhere to the untreated plate that well. So I've developed another method of washing the plates or removing the media from the plates. And it's kind of unusual, but you essentially invert the plate over a paper towel. And I don't know exactly why, but I've investigated it pretty thoroughly. And for our cells, at least, this is the way that causes you to lose the fewest cells, especially from the center of the plate. If you're going to do this, either do it under a BSC or ensure you're working in a BSL2 work environment. I'm going to play a video here that demonstrates how this method works. You'll notice I have paper towels set up here. You can't see it. Underneath this, I have plastic wrap. Since this is media from cells, you want to make sure you're doing it in a BSL2 environment. You want to be very careful about this. So I'm going to start the video now. So it's really weird when you see it, but somehow this is the best or the least bad way of doing this for our cells, at least. Now, one downside of this method is that the cells are exposed to air for a few seconds. In this video, I timed it. They're exposed to air for about 7 seconds, but a practiced operator can get that down to about 3. So depending on what your cell type is, you might want to try another way of doing this. In addition, in this example, you might ask, is contamination a problem? It's only going to be incubating for 37 degrees for an hour after this is done. In addition, our cells have unique phagocytic properties which lower the risk of contamination. So for our purposes, at least, I do not believe that this is a problem. So so once you've done that, once you've added the Seahorse media, you'll want to put that at 37 degrees in a non-CO2 incubator and allow it to incubate for 1 hour. In addition, this day you will want to add the calibrant to the cartridge. You'll want to remove the water from the cartridge and the accompanying plate and add the calibrant that I described. You can see me doing it there. You'll want to allow that to incubate for 45 minutes at 37 degrees Celsius. In addition, while this is incubating, this is a good time to dilute the reagents you'll be using for the assay. Depending on whether you're using the Mito stress test or the ATP rate assay, you will need to add, you'll need to dilute FCCP if you're doing the Mito stress test, and you'll certainly need to dilute oligomycin and rotenone AA to go with it. This is an example of how the reagents come. They come in a sealed, light-protected pack. They come in these little tubes. I don't know if you can see it very well, but down there at the bottom, there's some lyophilized FCCP. The advantage of purchasing the reagents from Agilent is that you can rehydrate them in Seahorse media, and you don't have to worry about any solvent or vehicle problems like you would if these were in DMSO. Interestingly, the kits that come from Agilent allow you to do up to 2 assays. So it actually has enough for you to do 2 Mito stress tests from one of these kits for ATP rate assays. So if you want to do 2 in the same day, you can do 2 on the Seahorse XFp during your reservation window, and then the kit will also have enough for 2 assays. Generally speaking, what I do is I'll have a Mito stress test kit, and then I'll do an ATP rate assay and a Mito stress test just from that one kit. Now, if you decide to do this, the substocks that you make up, you'll want to store them protected from light at 4 degrees Celsius. Do not store them at room temperature, and do not leave them in the trough like you see here. On the right, you will see a crude diagram that I've made up that shows you exactly how this works. It shows you the dilutions that you do. I'll provide this diagram to anyone who wants it. It helps to keep straight exactly what goes in what tube. So these are the master stock tubes like you saw in the previous image, and then these tubes are 1.7 milliliter tubes that you would have you'd have yourself, and you'd make up the media with those. Now, when you go to do the assay, when you go to the Seahorse room, you'll need to bring some of your own supplies. Specifically, you'll need gloves, you'll need a pipette, multi-channel, or otherwise, depending on what you're using, you will need pipette tips. And if you're using a multi-channel pipette like we do, you'll also need to bring a trough to go with it. In addition, you'll need to bring a flash drive to copy your data from the Seahorse XFp. The XFP stores data for about, I believe, 60 days before it automatically deletes it, and you'll need to take it with you. Certainly that day is advisable, so think of a name for it. I recommend, if you're not in Weber Hall, I recommend having a transport container to protect your plate from heat and light and keep it at the appropriate temperature as you're on your way to the stem cell core. Specifically, we've constructed this transport container you see here. It's very simple. We just have an old cooler from a previous shipment that came to us, and I lined it with this reflective material here. I believe this helps protect it from light and vibration, certainly, and then you close it. I also have these bottles in here. These are temperature ballasts. So what you can do is you put these at 37 degrees, let them warm up overnight. You put them in here with your plates, and that will help to keep the plate at 37 degrees Celsius on its way over to the stem cell core. In addition, once you on the way to the Seahorse room, once you reach the Seahorse room, you'll also want to have another trough with you or other container to put in the non-CO2 incubator to keep the humidity level high as you're working with your plate. So that's another consideration as well. And in addition, in our case, there's some extra space in the container for a multi-channel pipette tips, troughs as well. Now, once you reach the Seahorse room, you'll need to load the injection ports on the cartridge. The injection ports are these 4 components you see around here. This hole in the middle, this is where the sensor goes. Now, the way this works is that you will put the reagents that are meant to be injected into your mini plate into these ports here, and they're held in there by surface tension, essentially, in the media. And when the instrument needs to inject them, it will send a puff of air through whatever port is marked to be injected. So, for example, normally you'll add oligomycin to port A in each of these wells, and the instrument will send a puff of air and it'll get injected, and then it'll move the sensors up and down to mix it. Now, one important component one important thing to mention is that if one of the wells is not loaded properly, then the air will go through the unloaded well, and the other injection ports will not have their reagents injected. And I'll talk a little bit more about that in just a minute. So I have a video here. It's a little awkward because hang on a minute. It's a little awkward because I set it up to be filmed, but this colored liquid here will show you how you add it to the injection ports. So like I said, it's kind of awkward, but you can see it there, and then you'll notice as it gets injected, there we go. And that's kind of what you do with whatever it is you're adding at the time you add it. So this might represent oligomycin. So once you have loaded this accordingly on the instrument itself, you will start your assay, and it will load, and you'll put this cartridge in along with the utility plate you see underneath it. And the Seahorse XFp will undergo a calibration step for 15 to 20 minutes. Now, I'd like to mention, although the clear plate that you saw underneath this cartridge looks like a mini plate, this one right here, the one underneath, it is not. That is an empty plate that just has the calibrant in it that we discussed earlier. If you're doing this in the Seahorse room, the mini plate, which has your cells in it, is going to be in the non-CO2 incubator in the Seahorse room, and it's going to be sitting there awaiting the assay. So this is the cartridge, and utility plate is what is being depicted here. Now, one thing I'd like to mention is that when you're loading reagents into the cartridge, sometimes they can be loaded improperly. In particular, I'd like to draw your attention to injection port D on well E right here. And you can see in this case, rather than being loaded into the bottom of all the way to the bottom of the injection port, it's actually kind of adhered to the side. I did this on purpose for this demonstration, but it does happen as you're loading it with a multi-channel and a regular pipette, whichever you choose to use. So after you've loaded the injection ports, I highly recommend you inspect them to see if this has occurred. If this happens and you don't correct it, then the air will travel past this droplet and out this injection port, and it will not have sufficient pressure to inject the other reagents in there. Fortunately, this is an easy problem to solve. All you do is just stick a pipette tip down here, and that'll cause it to kind of drop into the bottom. Because these are held in with surface tension, you might wonder how easy it is for you to maybe bump this plate or accidentally cause the reagents to drip out the bottom. It's actually fairly difficult. The thing you want to avoid is sudden bumps, but that is not necessarily something to worry about too much. Now, there's also a question of whether you want to use a multi-channel pipette or just a single pipette. Some people prefer to use the single pipette because they believe it's more accurate. I prefer the multi-channel. It saves time, and I haven't noticed that much accuracy difference because of kind of the meniscus and the way these are loaded. It's sometimes hard to tell if these are at the same level or if it's just the way the reagent is oriented in the plate. So you can see here it's kind of curved. This one looks like it's maybe a little more full than this one, but maybe this end is higher. It's tricky to know for certain. But the good news is, because these injection ports end up empty at the end of each assay, you can actually keep them and you can use them for practice. So you can practice loading these before you even do the assay and get your skills up so that when it comes, you're comfortable and you know exactly what to expect in terms of loading accuracy and other problems as well. So once you have prepared the cartridge and utility plate, you will put it in the Seahorse XFp and you will run the assay. It will undergo that calibration step I mentioned. Then you'll take it out. You'll take your mini plate from the non-CO2 incubator, take the cover off. You will put it in the instrument itself. And then depending on what assay you're running, it will run for an hour to an hour and 30 minutes. The instrument will give you an estimated completion time, so you'll know when to come back. The Mito stress test takes about 30 minutes longer, so be sure to note that. In addition, I believe I should have mentioned, when it comes to loading the cartridge and utility plate, if you're using an expired cartridge, the instrument will note that the cartridge is expired, but it will give you the option to proceed anyway. So make sure that you go ahead and override and allow it to proceed. If you're going to do that, don't leave the room for 15 to 20 minutes for the calibration step, only to come back and see that it hasn't started yet, because that's a notification that you'll get. Now, while this is running, you can prepare for the normalization part of the assay. You will need to normalize to the number of cells that are in each well. There are a number of ways to do this: total protein, total DNA, and so forth. However, for our purposes, we prefer to use Hoechst to stain the wells. Now, one potential problem with this is that if you want to add Hoechst to a plate, you might very well be used to, say, removing the previous media from the plate, then mixing up Hoechst with your imaging media or PBS and adding it. I don't do this, and my reason for not doing it is because if I rinse the well after the assay, I might end up losing some cells after it's rinsed, and that could corrupt my measurement. So to solve this problem, what I do is I make up a Hoechst substock. Specifically, I add 1,080 microliters of Seahorse media to 2.7 microliters Hoechst master stock, and then I add 50 microliters to each well. So then this directly. So there's no rinse involved. At no point is the plate being rinsed after the assay is done. So that's good. And normally I incubate this for 10 to 30 minutes. Now there's a limitation here. Because you're adding a concentrated Hoechst mixture and it's diffusing through the well rather than just being consistently added at a consistent concentration, your staining intensity can be kind of different from one well to another. So be aware of that. We found a way to correct for this computationally, however, and I will describe that in just a moment. What we do is we take images of each well. We take a 4x4 tile scan, so that's a total of 16 tile scans, and we have Z planes within each of those as well. So we're taking multiple Z planes within that 4x4 tile scan. That's a total of 16. So once we take these images, I have developed a custom MATLAB script to analyze this. Now, before we proceed to talk about that process, I'd like to draw your attention to one particular thing. I'd like you to note the presence of these three, I call them dimples, on the bottom of each well. These are present in both the utility plate, which has the calibrant I mentioned earlier, as well as the mini plate, which has your cells in it. Now, these dimples are necessary to support the sensor in the Seahorse cartridge to stop it from touching your cells at the bottom. However, if you're using the method that I'm describing, these can constitute a problem because they're curved. They're like a little dome. So they sort of magnify and distort the cells that end up on them and end up stained with the Hoechst. So there are a couple ways of dealing with this. The thing I'm doing is basically I'm just deleting them to avoid having them corrupt the image. So essentially, our normalization process works like this. We stain the well. Based on the total intensity of this, the script automatically determines a cutoff for what constitutes stained nuclei and what constitutes background stain or something like that. And then it does a kind of binary image that you see here. So you can see how these are nuclei, kind of these yellow structures here. But here in the dimple, you can see how, because of the curved nature of it, it distorts the image. So our script finds the center of the well, and then it based on that, it knows it can then infer the location of the dimples, and then it just deletes them. So you are losing the cells in the dimples. Those are not being counted. That's going to distort your cell number slightly, but I reasoned that this is the least bad way to deal with this problem. And I'll talk about some future directions that will be even better a little later on. So here you can see the raw image of a section of the well. You'll notice some debris here. The primary cells we use tend to produce debris that can be stained by Hoechst. I've tested it. It's not mycoplasma or contamination. It is just debris from these primary cells that are producing a lot of it. In any case, once the threshold for nuclei is obtained, as you can see here, we have a problem. We need to know the number of cells in each well. But how can we do this? Because of the density we have to seed cells at, as you can see here, sometimes the nuclei touch each other or overlap. I don't like this, but because our cells are not that active, we have to seed at a very high density to be within the appropriate reading range of the instrument. So we need to count these. We want an absolute cell number. But because the cells are touching each other, we can't just count the number of objects. For example, this large formation here would be counted as one nuclei, even though it's quite clearly many. So what the program does is it eliminates anything that is too large to be one nucleus, that is large enough to be a nucleus, but small enough to be just one nucleus rather than one of these multinuclear formations. And you can see that in the comparison between these two images here. Once this is done, the program finds the average intensity of each of these, and then it comes up with a mean intensity of each nucleus. Then it identifies all of these objects and divides their total intensity by that mean to produce a number. And you can see that here. So you can see it works fairly well. Mostly you're getting individual nuclei. You can see this is quite clearly 2 nuclei. This is quite clearly 3. This is about 6, but this is more like 3 nuclei rather than 4, and that's definitely just about 2. So the script isn't totally perfect just yet. There are some quality control outputs in the script as well that will help you with this. It will generate histograms and plots to tell you if you have any outliers or how many objects you're getting that contain how many nuclei, things like that. This script is kind of a project for us in and of itself. In the future, I hope to apply machine learning to distinguish individual nuclei. This seems to me like a fairly straightforward application for that. Interestingly, I haven't found any commercial method that can do this. But this is what we use, and I think it's pretty good. And I'd be willing to share this with anyone at the talk today and give you some guidance on how to use this. So what the script does for each well is it adds up all the nuclei that are in here, and we get integers, numbers of cells in each well. Now, once you have those numbers, you can enter them into the Wave software, which is supplied by Agilent. You can freely download it. You tell it how many cells are in each well and give it a scale factor. Then once you've done that, a few things you'll need to know about the software. You'll need to add a new media, and then you'll need to select the media that you're using. If you're like us, you'll be using the Seahorse XFp media at the pH we described, the buffer factor as well, and the other components. You've added the glucose, the glutamine, the pyruvate. Then you'll need to go through here for each of the categories. We only have 2 here, and you'll need to select what media you're using. So make sure it's assay media 1 to match this. Otherwise, it won't work. You'll also want to check the normalization box. And then this will give you OCAR and ECAR per cell, which we discussed in the previous lecture. However, if you want more detailed readings, and in particular, if you want absolute ATP levels, you'll need to export it via this report generator, and you'll select whichever one fits your assay. So if you're doing the ATP rate assay, and that's what this is, you'll select this, and then you'll open basically an Excel document, and it will give you all of the information that we discussed in the previous lecture, all of the details in terms of ATP production or OCAR readings and Mito stress test parameters, if that's what you've decided to do. I'll mention a few other notes. When it comes to using the Seahorse XFp, you don't have to use the assays they mention. You can actually tell it to inject whatever you want from whatever injection port you want at whatever time you want, and you can tell it to take measurements whenever you want. So it's actually better to think of the XFp as a pH and oxygen sensor that can also inject stuff into your mini plate whenever you want. And so you could use it to do something completely different or creative with your cells if you wanted to measure pH and oxygen consumption in response. In addition, there's a new assay from Agilent called the Mito assay, which uses oligomycin and FCCP only. This just came out, so I'm not familiar with it yet, but it is something that exists if you want to study toxicological aspects of mitochondrial performance. You want to see how performance is being impaired in that way. Now, before I move on to the Q&A, I'd like to answer what I suspect will be some questions. One question, can I reuse the cartridges? That's the thing with the injection ports and oxygen sensors. No, you cannot. They are one-time use. Once the fluorescent reactive oxygen and pH components have reacted, they've been used up and you can't use them again. You might ask, can I use my own reagents, oligo, FCCP, rotenone AA? Yes, you can. I have tried using our own reagents that we've made up. So we just ordered all those components in bulk from a supplier, not from Agilent. I have not been able to detect any difference. So here, if you do an ATP rate assay, it's $55 if you buy a brand new one from Agilent or from Rachel in the Stem Cell Core. But if you really wanted, I could just lend you the oligomycin and rotenone AA that we have. It's much less expensive just for one run if you wanted to skip this. I'm not sure how well they last at -80. They seem to last a long time, but I can't vouch for that. In addition, you might ask, can you use expired reagents? I've tried using expired reagents, including ATP rate assays and cartridges. I have not been able to detect any significant difference. Certainly, based on the response I've seen, if you just want to practice this, definitely you can use expired cartridges and mini plates. These are available from Rachel in the Stem Cell Core at a reduced cost, so you can talk to her about that. You might also ask, can I put things other than adherent mammalian cells in the mini plate? Yes, you can. I believe you can do suspension cells. I think you can put C. elegans in there. If it'll fit under those dimples I mentioned, you can measure it in the XFP. If you have larger like tissue samples, there's a Seahorse XF24, which can hold those. That's available at Loma Linda. I believe you'd have to contact them and they might let you use it. So you could do tissue samples or samples of larger cells that way. Depending on how big your cells are, though, and depending on how metabolically active, it might be worth trying this. You could at least get a mini plate and try seeding your cells on it for practice. You don't have to necessarily try the whole assay all at once. If you just want to try culturing the cells on a mini plate, you can order the mini plates from Agilent, and it's a little cheaper and can work well just for practice. You can practice your rinsing, staining, normalization, whatever you want. And I'd like to conclude now by opening it up to questions if anyone has any.
Thank you, Shane. That was a great presentation. I do have a question about the normalization. So you mentioned one way, whatever you are using, right, the Hoechst stain. But is there any other method that you know of?
I have never used any other method. However, there have been people on campus that have used total protein content. You could do total DNA content would be another way. You might be able to do total Hoechst intensity, which is a variant of this. Those are the only methods that I know of, but other methods do exist.
Okay, and you recommend this because this, like most people use it, or you have standardized this for your cells?
In terms of the Hoechst method, I don't know what the most common normalization method is overall. However, one of the things I observed recently is that a lot of publications have people doing absolute cell number as determined by Hoechst staining and counting. That's very popular. In addition, because these cells that we're using have not been metabolically characterized very well, we are interested in learning what their metabolic activity is per cell, in an absolute sense. So this is what I've optimized for my cells, but I would hope that we have a good enough protocol to where you probably could, if you have adherent mammalian cells, you probably could just do the same thing more or less without having to go through the optimization process or without having to develop the same optimization process that I've developed.
Okay, thank you.
Are there any other questions? No? All right, then. I'm going to go ahead and stop the recording.